Hemostasis is the process of blood clotting in order to prevent blood loss when vessels are damaged. This process has three different phases: the vascular phase, the platelet phase, and the coagulation phase, and it ends with clot retraction. In this video, we're going to walk through each of these phases to show how our body helps prevent major bleeding.
The vascular phase lasts about 30 minutes and involves the contraction of the blood vessel called a vascular spasm, which decreases the diameter of the vessel and slows down or completely stops blood loss. Here are some of the changes that happen in the endothelium of the damaged vessel. The endothelial cells contract and expose underlying basement membrane to the bloodstream. The endothelial cells release chemical factors and hormones. The plasma membrane becomes sticky.
The platelet phase begins when platelets attach to the sticky endothelial surface. As more and more platelets stick to the surface in one another, the platelets form a plug. This process is called platelet aggregation. The forming of this plug begins within 15 seconds after injury. The platelets release a variety of compounds including ADP, thromboxane A2, serotonin, clotting factors, platelet-derived growth factor, and calcium ions. These compounds produce a platelet plug that will be reinforced as clotting occurs. There are many different factors that help keep platelet aggregation controlled to the specific injury site.
The third phase of hemostasis is the coagulation phase. This is also known as the blood clotting phase. The clotting factors, or procoagulants, in the plasma are important to initiate normal blood clotting. Here is a chart that shows the many different clotting factors involved in the clotting process. The extrinsic, intrinsic, and common pathways are involved during the coagulation phase. The extrinsic and intrinsic pathways converge at the common pathway, which stimulates the production of fibrin, which then forms a network that traps the blood cells and platelets and helps form a blood clot. This acts as a seal for the damaged portion of the vessel.
Clot retraction is when the damaged edges of the vessel are pulled together. The damaged area's size is reduced, decreasing the bleeding and allows for other cells to complete repairs.
Fibrinolysis is the final step in the process. This is where the blood clot dissolves. It begins when thrombin and tissue plasminogen activator activate the proenzyme plasminogen. This stimulates the production of plasmin, which eats up the fibrin strands and erodes the clot. After this is completed, we're left with a healthy vessel once again. This is hemostasis.